Good morning, everyone. A very warm welcome to all the participants joining us today for the webinar organized by Indian Pharmacologists United Network. I am Dr. Harsh. I will moderate today's session. It is our privilege to introduce Dr. Vipul Chaudhary, sir, a distinguished academician and researcher in the field of pharmacology, who will delivering today's webinar on from inhibitor to targeted protein degraders, a new paradigm in drug discovery. Sir currently serves as professor and head of the pharmacology department at GCS Medical College with over 16 years of rich academic experience in teaching, research, and academic leadership. Bridging academia with industry, he has worked as a principal investigator in Lambda Therapeutics, where he was actively involved in phase 1 clinical trials and BA/B studies for nearly 3 years. Sir is also a member of two ethics committee contributing to ethical oversight of clinical research. In addition, he has delivered numerous guest lectures at state and national levels on key areas such as clinical practice and pharmacokinetics, inspiring student and professional life. Before we begin, a quick announcement. Feel free to drop questions in chat box. We will address them during question and answer segment. With this, I invite Dr. Vipul Chaudhary to begin his presentation. Over to you, sir.
Hi, good morning to all. Am I audible?
Yes, sir.
So, before we start, the topic is now we have the new protein degraders, which we are going from inhibition to the targeted protein degradation. That is a new paradigm in drug discovery. And we have two molecules, that is PROTEC and molecular glue. So let me tell you, before we go into the PROTEC and molecular glue, let's see why actually we require these two proteins. So first, we'll see what are the limitations of the small molecule, that is current molecules. So as we know that the current molecule, the drug molecules, are only inhibiting the protein and not eliminating. Suppose if I give the example of ACE inhibitor, what we are doing? We are just inhibiting the ACE. Like beta blocker, we are just blocking the beta receptors. So what happens, as long as the drug is present on the receptor, actually the effect persists. And when you take away, like suppose if you stop the beta blocker, again the BP can increase. So it requires a continuous target occupancy with a small molecule. The third issue, why we are talking about the protein, let me tell you that because all the receptors, all the enzymes, all the channels, they are nothing but the proteins. So these ACE inhibitors, beta blockers, sodium channel blockers, we are actually blocking the protein. Okay. So the third issue with the small molecule is it can only target the protein with an active site. Now, before we go to this, what is the meaning of this? Now we know that the drug to act, the drug has to bind to that receptor. And for that, it needs binding site. That is called the druggable protein. But there are various proteins like a transcription factor. I hope you know that transcription factor is nothing but a DNA binding protein, and it's regulate the transcription gene. So this transcription factor, scaffold protein is nothing but it brings all the multiple protein together for the signal generation. So this transcription factor and the scaffold protein, they are very huge protein and they don't have any groove in them where actually your drug can bind. So your drug cannot bind to such factors, and also their surface is very flat. Actually it requires the groove, but the surface is very flat, and these are the very large protein. So your small molecule cannot inhibit the entire protein. And we know that this transcription factor and scaffold protein, if there is any issue, it can lead to the cancer or any inflammatory conditions. So the third issue is those drugs cannot inhibit such a huge protein. The fourth one is resistance due to mutation. Like example I have shown here, EGFR, growth factor inhibitors. Actually, it is a large molecule with around 210 amino acid sequence. In that, if there is a, 1200, and if there is any mutation at 790 position, where instead of threonine there is a methionine, what happens? This drug, gefitinib and erlotinib, cannot bind to EGFR and cannot inhibit that. So resistance due to mutation is also one of the problem with the small molecule. Obviously, we know the off-target toxicity, that not only the main target, it can also lead to the side effect if the drug level is increased. And the last is cannot remove the non-catalytic or scaffolding function. Now, what is this non-catalytic and scaffolding function? So any protein has, some of the protein has two type of action. It has catalytic activity, means it performs a chemical reaction, like kinase. Kinase has phosphorylated activity, which gives a phosphate. And what is the scaffolding function? Scaffolding means it acts like a platform. And in this platform, other protein comes, they bind and causes the action. So it's like it is providing the platform for other protein to act. So this, with this small molecule, even if you remove the catalytic activity, its scaffolding function will still persist. I have given the example of focal adhesion kinase, and it provides a platform for integrin and growth factor to generate the signal, helps in the cancer survival cell and migrate. So these are the limitations with the small molecule, that this drug only inhibits the protein, only inhibits the catalytic activity, and they cannot act on the protein which has mutation. So now the scientists are coming up. Now this PROTEC and molecular glue are still in the infancy stage. So scientists are coming up with the idea that why not to degrade the protein instead of just inhibit. So that's why they have developed the molecules like a PROTEC and glue, which will degrade the protein and not only it inhibits. So I hope I am clear before I go to the PROTEC and molecular glue. Any participant, any doubt till now? Participant can unmute themselves. Any doubt? Am I audible? Because
Yes, sir, you are audible.
speaking, there will be two possibilities. I am not audible or they have not done anything. So I hope they have understood that what is the limitation
Yes,
of
sir.
small molecules. Now, before I go to the PROTEC and molecular glue, let's understand how actually protein get degraded and how actually the PROTEC helps in this protein degradation. So if we see the protein degradation, it is by proteasome ubiquitin process. This is normal physiology. So whenever there is a damaged protein, actually in our body the proteins are continuously degraded. As if damaged or if its function is over, or any misfolded protein, they are continuously degraded in the body by proteasome. So what happens? The first, we have enzyme E1. So ubiquitin will activate the E1 by the ATP, and this ubiquitin will bind to the E1. This E1 further with the ubiquitin will be activated by E2, and this ubiquitin will be transferred from the E1 into the E2, and this E2 will go to the E3, which is conjugate protein. Now, what is the role of E3? Now, it is very important to understand here. E3 will recognize the damaged protein. Okay, so I have given the name substrate. E3 will recognize the damaged protein and will carry this E2 near to the damaged protein, and this E2 will transfer the ubiquitin onto the substrate or damaged protein. This process will continue till there are more than 4 to 5 ubiquitin present on this damaged protein. This is called polyubiquitination. Now, proteasome, which is one of the machinery to degrade the protein, will recognize this protein which has got 4 to 5 ubiquitin. So it's like, suppose if I give you very routine example, you want to kill one of the terrorists from the crowd. What you will do, if you know that somebody is terrorist, what you will do, you will put some type of cap on that terrorist head, 5 to 6 cap, and police or the shooter will recognize such person and he will kill them. So proteasome will recognize such damaged protein with this polyubiquitination and degrade the protein. So this is a normal process how the protein is degraded in our body. So now, what is PROTEC? The name is proteolysis targeting chimera. It targets the protein, lyse it, and chimera means, it is a Greek word. Chimera means the animal which has got more than 3 different heads. So if you can see here, can you see the cursor I am showing? So it has got 3 heads. One head is with E3 ligase recognizing moiety. Another hand which binds with the ligand or the target protein which we want to destroy. And these 2 hands are bind with the linker. Okay. So it is like he has 2 hands, like a person which has got 2 hands. One hand with bind to the E3 ligase and another hand with bind to the target protein. And this person will bring E3 and protein together so that E3 can ubiquitinize the target protein. So PROTEC role is nothing but just to bring, here you can see E3 and target protein are together. So then only this E3 with the can transfer the ubiquitin from E2 to the target protein. So PROTEC will have nothing but the role of bringing this E3 and substrate together by attaching. So first it will attach to the E3, again it will attach to the target protein and bring both the protein together, both the E3 ligase and protein together. So E3 can put the cap like a ubiquitin on that, so proteasome can destroy it. Okay, so this is the role of PROTEC, and PROTEC is not a simple molecule. It is made up of chemicals. So it's not that simple as I have drawn. Here I have shown the 3D picture of how the protein looks. This is a E3 ligase. Blue color is target protein, and in between you have protect with linker. This is another diagram where it is showing the E3 ligase target protein binds with the protect. This is a protect in between. Here is a protect, another protect. Okay. So if you see the in animation, this is E3 ligase. This is your protect, and this is your target protein. So it binds this E3 and green color target protein. You can see the small protect here, and this is ubiquitin. It will keep one ubiquitin here, again goes back, bring back another ubiquitin, again attach, again goes back, and that's how it will polyubiquitinate the target protein, and this protein will be recognized by proteasome, and it will destroy the target protein. So this is a mechanism of protect. But what are the challenges when you prepare the protect? It is not that simple as it looks. So let's see what are the challenges when you prepare the protect. Number one, large molecular size, more than 800 to 1000 Dalton, and that's why it has got poor cell permeability. Now you know that as per the Lipinski rule, if molecular weight is more than 500 Dalton, it cannot enter into the cell. And remember, this proteasome and E3 are present inside the cell. So recently, the most challenge is how to enter this protect inside the cell. So it is difficult, or I can say it has got poor cell permeability. The second is E3 ligase. I told you that it will bind. E3 is the main ligase which recognizes the protein and destroy. Now, more than 600 E3 ligase is present in our body. Out of that, only 3 to 4 E3 ligase structure the scientists have recognized. So unless you will not recognize the structure of E3 ligase, you cannot develop the protect. So out of 600, only 2 to 3 or 3 to 4 E3 ligase like cerebellum, von Hippel-Lindau, VHL, mouse double minute2, MDM2, and few less RNF114 have been identified. So unless we will not identify, the structure of E3 ligase, we cannot make the protect, and we cannot destroy all the proteins which are causing the cancer. So limited E3 ligase diversity. Another is optimization of linker length and composition. Now there are two things: composition and linker length. First, let me tell you why linker length is important. So let me go back to my slide. This is the linker length. This is the length of linker, and it has got two hands. So let me come back to the previous slide. Suppose this substrate and E3 ligase has to come together, then only this E3 will put the ubiquitin on this protein. Suppose this is a protect, and these two are the E3 ligase and target protein. Suppose you have to put a cap on someone, and if that person is very far from you, your hand will not be able to reach. So if the linker length is too large, E3 will not be able to transfer the ubiquitin on the target protein. So number one, that length should not be too large. And second, it should not be too short. Suppose that person is almost touching you and standing, and you have to now put the cap. It is very difficult because your hand will overshoot his head. So optimization of linker length is very important of that protect. So then only E3 and will ubiquitinate the protein. And the another is composition. This chain is made up of different structure: PEG, alkyl, triazole, piperazine. Earlier, alkyl were used commonly, but the issue with the alkyl was it has too much of hydrophobicity. So the cell permeability was less. Mixing alkyl with the PEG, yes, they improved the uptake of cell, but affected the PK. And also it is too flexible. Now when your hand is too flexible and you have to hold two people with your hand, and if your hand is too flexible, these two people, the E3 ligase and the substrate will not become stable, and the ubiquitinous process will be tough. So now the scientists are using the more rigid structure like triazole, piperazine, and piperidine, which gives this ternary complex a stability. Ternary complex means one side E3, in between protect, and other side is target protein. This ternary complex becomes stable, so the proper ubiquitination can occur. So challenge is for the length and composition. Another is off-target degradation. Now, let me tell you, it is not that simple. How you prepare the protect? So you first have to study the structure of E3 and you make one hand of protect. Then you study the structure of target protein and you make the another hand. Now, if any changes occur with either of the hand, it can bind to the other good protein and can destroy the good protein. So off-target degradation can occur if your chemical composition is not proper. Another is hook effect. What is hook effect? Now you know that this is a proper ternary complex. This is E3. This is protect in between, and this is target protein. Now, if your concentration of protect, amount of protect is normal in your body, it will bind to both the E3 and target protein in a proper composition and makes the target protein. Makes the ternary complex. But suppose if you give too much of protect, what will happen? Now this protect will bind to E3 ligase, another protein will bind to the target protein, and it will not form proper ternary complex because now the body has excess of protect. So now protect will bind to the separately the E3 and target protein and forms a binary complex like this. So up to certain level, your ternary complex formation is proper, but after that, the target degradation will decrease because in binary complex, there is no degradation of protein because either E3 is absent or target protein is absent. So it has to have proper ternary complex. So that's why it is called hook effect. And of course, last but not the least, this is not the simple small molecule whose structure we know, so we can make the generic drug. No, this is a protein. And depending upon the E3 and target protein, we have to make the proper protect. So the complex design and manufacturing, its PK, PD parameter, we have to take care. So it is very difficult for the scientists to develop. So these are the few challenges which the scientists are facing while developing protect. Now coming to the application of protect. wherever the protein is present, we can use this protein. Like neurodegenerative disease, we have target protein like tau, synuclein, mutant Huntington's, which are responsible for neurodegenerative. We can target in immunotherapy. We have STAT, JAK protein. We have tyrosine kinase 2. In cancer, we have BRD4, BTK. So all this protein mutation causes cancer, immunotherapy, or neurodegeneration. In bacteria and parasite, we have different proteins which are responsible for their growth. In cardiovascular, we have PCSK9, BRD4, which are responsible for either cholesterol, LDL cholesterol regulation, like PCSK9. AMPK is a cellular energy. It senses the cellular energy and responsible for glucose uptake, lipid synthesis, and protein synthesis. So any mutation in this viral protein, these are all proteins which are responsible for the viral survival and replication. So all these proteins we can target with the protect. We can prepare a protect as per this protein structure, and we can bring back near to the E3 and we can destroy. Now, what are the different strategy in protect design the scientists are bringing? So the limitation of the protect can be inhibited, or we can overcome this. One is photo switchable protect. See, these are the only the thoughts they are implemented. Okay, it is in the infancy stage, so we have not still developed, but this can be the few thoughts which can be helpful while designing the protect. One is photo switchable protect. Now, whenever you give protect, if it goes to the other protein, or if it goes to the other cell, or if we want the specific protect to be activated when it reaches to the specific site, then we can develop the photo switchable protect, where there is a azobenzene ring is present, and this azobenzene ring will be activated once we give the light, UV light, when this protect reaches to the proper target. So one is photo switchable protect. Another is photo case protect. So one of the either E3 ligase or target protein hand is caged or inactive by nitrobaratryl oxycarbonene. These are the few chemical group which are light sensitive. You can cover with this, and whenever the protect reaches to the proper site, you give the specific wavelength of light, and this cage will remove and it will become active, and that part will bind to the particular protein or E3 ligase. Third is clickable protect. Now I told you that protect is a large molecule. So what to do? We can make two pieces. We can prepare in two pieces, and both the piece has TZ, tetrazine and trans-cyclooctane chemical group at the end. These two pieces, small pieces, will enter inside the cell, and then these two chemical will bind and make large protect inside the cell. So one of the limitation of the large molecule which we saw earlier can be overcome. So this is a clickable protect. Another, we can target the multiple protein. So one side we have E3, and other side we can give two hand which will bind to two different target protein, and we can destroy the multiple target protein at one go. Another is antibody or small molecule conjugate protein protect. So one side we have E3 ligase, another side we have antibody conjugate protect, which will take us to the targeted antigen, will bind, and this E3 will ubiquitinize that protein. So it will just the proper target delivery protein. And to kill the transcription factor, we can bind another end with the oligonucleotide, which will take us to the specific target like a transcription factor, which are mutated. And actually the portion of the DNA can be destroyed. So these are the just the thought the scientists have, that such design we can incorporate while preparing the protect. Now this is the homoprotect. Both the side is E3. Both the protein, both the target protein, like, okay, let me give you the example, MDM2. This MDM2, actually the function of this MDM2 is to control the P53 transcription factor. This P53 actually regulates the cell division. So if there is any mutation in P53, or if P53 is inhibited, the cell will keep on divided and cause the cancer. Now this MDMT2 keeps a check on P53. Whenever the proper cell division is occur, or it will kill the P53, otherwise it will allow P53 to act properly. Now if there is any mutation in MDMD2, it will unnecessarily kill the P53 and cell will keep on divide. So this MDMT2 is itself act as a E3, and if any mutation occurs, it becomes a target protein. So what we can do, we can bind MDMT2 at both the sides. So this MDMT2 will transfer the ubiquitin onto this MDMT2, and this MDMT2's ubiquitin will transfer to here, and both will be degraded. So this is called homoprotect or suicide cleavage. So these are the few strategies we can implement while protect design. So what is the advantage of protect over the small molecule? It has got catalytic mechanism. What is catalytic mechanism? It acts in a slow, lower the dose. So once we give protect, it brings E3 and target protein and it destroys. Again, protect will release from that and bring back another E3 ligase and again destroy or target the protein which is damaged. So lower dose, continuous effect, that is called catalytic mechanism. Second is complete removal of protein. It is not only inhibiting the protein, it is removing the protein. So complete removal of the protein. Third, if protein has no groove or flat surface or large protein, that can also be targeted with the protect, reduce the drug resistance, and effective even at the protein is at the high levels. Because our drug, the small molecule, if the protein is very large in number, it cannot cover entire protein. So this problem can be overcome by the protect. So overall, PROTEC is nothing but it uses the normal human machinery, which is actually degrading the protein, and it is bringing back the E3 and the normal protein together and just helping them for the ubiquitination. So PROTEC is just using or hijacking the normal proteolysis machinery. So what are the PROTEC in the development stage? We have few PROTEC which are in the phase 2 and phase 3 trial for like ARV-471. It degrades the estrogen receptor in ER positive breast cancer, and ARV-110, which is androgen receptor degrader for the prostate cancer, and other is for the lymphocytic leukemia for the by targeting BTK. But it is still in the development stage, and we'll have more idea when it develops and comes out in the market, that how it behaves, for how long it acts, and what is how long it acts, and what is the time required for the protein to stop the functioning. So all these things will have idea once it gets developed. So now this was about the PROTEC. Coming to the molecular glue. Molecular glue is nothing but similar to the PROTEC, but the difference is, instead of the 3 hand, the molecular glue sticks the E3 ligase and target protein together. So let's see what is the history. That thalidomide, how we got the idea of molecular glue. So we know the thalidomide, which was used in 1950 as a sedative and morning sickness, but caused the severe birth defect. You know why it caused the severe birth defect? Because molecular glue targeted or attached with the E3, and it attached the SALL4 protein. This SALL4 protein is responsible for the limb development. And what happened when we gave the thalidomide, it brought E3 and SALL4 together, and our E3 destroyed the SALL4, and that's how our limb could not develop. So later it was found that thalidomide was also acting as a molecular glue, and we have developed some of the glue which bind the CRBN. I told you one of the E3 ligase, only 3 to 4 E3 ligase we have identified, and one is CRBN, cereblon, and it has degraded the IKJF1, which is important for the survival of myeloma and leukemia cell, and that's how we are using it in the multiple myeloma. So all these thalidomide cogners like lenalidomide, pomalidomide, now we are developing as a molecular glue. So what is the mechanism of action of molecular glue? This molecular glue has different action than the PROTEC. In PROTEC, first we used to study the E3 target protein, and we used to prepare 2 hands of PROTEC, and we used to bind. Now this molecular glue, what happens? It binds with the E3 ligase and changes the surface of E3 ligase in such a way that it binds with the target protein. Now here, can you see this groove in the red box? It is a normal E3 ligase. This black is the molecular glue. When it binds with the the E3 ligase, it has changed the surface of E3 ligase in such a way that now this E3 ligase will go and bind to the protein and destroys that protein. So this is actually, we have not developed the molecular glue, we have got by the serendipity in form of thalidomide. Here you can see the protein structure. This CRBN is E3 ligase. This is CK1 is our target protein, and this is a molecular glue which has changed the surface of CRBN in such a way that now it will bind with the CK1A. This is the glycine backbone of E3. This is E3 molecular glue, and this is a target protein. So when we found that thalidomide is one of the molecular glue, what we did, we studied the structure of thalidomide, and from there we made some few changes in thalidomide and prepared the new drugs or new molecular glue like lenalidomide, pomalidomide, and now we are still developing few more like NRX-1532. So what is the difference of PROTEC with the molecular glue? If you see the structure, PROTEC is bifunctional molecule which has two hands and in between it has linker. It is monofunctional. It binds only to the E3 ligase or surface, the target protein, and makes the changes. So it bridges the E3 and protein, it induces the changes into the E3 ligase. Another is development. The rationally we can develop, but here it is very difficult. I will tell you why it is very difficult. Okay. E3 ligase uses a known E3 ligand. It can alter any E3 substrate specificity. So flexibility is, yes, we can redesign. We have less flexibility. I will explain these three in the limitation. Example, you have seen ARV 110 and 475. Here thalidomide, lenalidomide, these are the few examples of molecular glue. Now, let me tell you why it is difficult, these three. Suppose, as I said, molecular glue we have not developed. What is molecular glue? Molecular glue is such molecule which goes and binds to the E3 and changes the surface of E3 in such a way that this E3 will bind to the target protein and destroys. Now, when we don't know that which type of changes will occur on the E3, how can we are sure that which protein, or the target protein or the cancer protein will be attached to that E3 or not? So we don't know where glue will actually binds onto E3. Even if it binds, what type of surface rearrangement it will bring onto the E3, and whatever the E3 ligase surface it has bind, whether now this E3, new E3, will be able to bind to our target protein or some other protein, we don't know. So now you know. Now you know how difficult it is to prepare a molecular glue. And that's why I said it was by serendipity we have found that thalidomide was acting as a molecular glue, and now we are preparing a new molecular glue with slight change with the thalidomide structure. So why we are doing, why we are preparing the molecular glue when we have protect. Only thing is, it is very small molecule. So our cell permeability issue can be taken care. So this is all about the protect and molecular glue. I hope you have understood. And that is the end of my session. Now stage is open for any doubts if you have. Thank you all. Yes, any doubt? Should I stop the sharing the screen, or you want me to keep?
Yes, sir. You can.
Yes. Doubt, please. How many? I can see the 34 participants. Have you understood all? Harsh, you can unmute the participants.
Yes, sir.
Sir, thank you for nice presentation. Very difficult topics made so easy. And sir, can you explain in the third slide what is off-target effect of protect? The third slide probably, or
Yeah.
non-catalytic action.
Yeah, what is off target? Now, when I showed the structure of protect, you know that we have prepared. It's like, can you see my two hands? Suppose I am a protect, this hand is for the E3 ligase and another hand is for the target protein. So, by mistake, because this protect is entirely nothing but the chemical structure, by mistake, if there is any chemical modification and instead of our target protein, by mistake, if binds to the other protein, then that protein will be degraded. So that is one of the disadvantage with the off-target effect.
Okay, thank you, sir. Thank you.
And it can happen to the E3 ligase. I mean, suppose if there is any chemical structure change, it will not bind with the proper our required E3, and it will bind to the other E3. So both ways it can happen.
As of now, there is no drug in the market, only in the phase 3 trial.
See, let me tell you, one drug, ARV-110, is in the development stage. I mean, it's a front runner. I could not find any much PK and PD characteristic of the drug. Actually, I was searching that once the protein is degraded, how long the effect persists, and when this protein gets degraded. I could not find, but one thing to find is that A110, that is, I told you, estrogen. It has T half-life is 110 hours, and it has been given in the 420 milligram QD. Now let me tell you one. Dr. Neela, I mean, your voice is disturbing. Now, let me tell you, when I told you that protein, once it degrades, it's not that it will not come back. Protein, our body, the proteins are keep on degrading, and it again, new proteins develops. So when we inhibit the protein with this protect, the only advantage with protect is your half-life is increased. As long as that protein, the new protein will not come up, you don't have to worry about the second dose. So when you give the second dose, again, it will inhibit the protein. So compared to the small molecule, here your half-life is increased.
Okay, okay. Thank you, sir. Thank you.
Thank you, Dr. Tanmay.
Sir, there is a one question in chat box.
Yes.
A similar question like off-target degradation by molecular glue.
Okay. So I hope I am clear with that.
Yes, sir.
Any other question? No question.
I think, sir, no
Okay.
doubts.
Okay. So thank you all for your patience in hearing.
That's bring us to end of the today's enlightened session. On behalf of IPUN and entire organizing team, I extend heartfelt thanks to Dr. Vipul Chaudhary, sir, for sharing such a detailed and practical perspective in today's session. I would like to thank our founder and coordinator, Dr. Arvind Singh Panwar, whose vision to creating IPUN brought us together today. Also, like to thank Dr. Anupama Desai, Head of the Department of Pharmacology, SMIMER, and also Organizing Secretary, IPUN, for unwavering support. Thank you all the participants for joining for today's interactive session.
One more doubt I just got. As a PG student, what can we do to this? Yes? Somebody asked.
Yes, sir.
Okay. Let me tell you. This was more for this topic or this protect design is more of the chemical changes. When I was going through this protect literature, I found the person who was developing the protect. They are from Bangalore. I mean, there must be many who are developing, but one of the person is in the Bangalore. They are working on this molecular glue and protect. So actually, the work is from the chemist side. So as a pharmacologist, the clinical, I mean, we are more on the clinical pharmacologist. We cannot, I mean, we should have idea about the chemistry part and how the protein docking part. So development part, this is not our domain. Our domain is for on the clinical side.
As a PG student, I think one is more important that it can come in the disadvantages question. Is a good
Yes,
question for
yes,
recent.
yes. Yeah, so my purpose of taking this was that you should know that such type of drugs are coming up. You know, it was very difficult for me also to understand such protein because protect and molecular glue was entirely the chemical part. So, which you saw the diagram, you know, which you saw was also provided by that person when he was explaining to me. So it is very difficult, but only as a MD pharmacologist, we should know that such drugs are in the development and it is coming up, which will remove the issues of the protein which are undruggable with our latest drugs.